Good morning. I'm Marty Downs. I run the LTER network office. I'm excited to hear from two of our current LTER working groups this morning, but first I want to do a quick overview of our active request for synthesis proposals. The LTER network office fosters enhanced collaboration, synthesis, training, and engagement across the LTER network. We are hosted at NCEIS, which has over 30 years of synthesis experience. LTR sites have more than 46 years of data, mainly hosted in the Environmental Data Initiative repository and focused on our core research areas, including human-environment interaction. We welcome efforts to combine LTR data with data from other environmental networks and individual researchers. In 2026, we are soliciting proposals for full working groups running 2 to 3 years with LNO data analyst support. The groups we'll hear from today were funded in 2025 as shorter-term Spark working groups. A bit more here on the difference between the two funding models. When proposing full working groups, you should have a committed team and have done enough legwork to be pretty sure your analysis is going to work. Spark groups are intentionally shorter term and more exploratory. We are, of course, most interested in proposals that draw on LTER's primary research areas of primary productivity, organic matter movement, nutrient cycling, populations, disturbance, and human-environment interactions. In 2026, we are especially interested in proposals that are responsive to our recently released strategic plan, especially including efforts to forecast responses to conditions that haven't previously been encountered, and efforts to connect ecosystem science to management needs. Some key dates for the active RFP. We have a nice long open period this year for developing proposals, with proposals being due on September 16th, 2026. We're expecting selections to be made and projects to start by late 2026 or early 2027. If you're interested in developing a synthesis proposal, definitely visit the LTR website for links to additional resources. And with that, I'll turn you over to our main event. Kai Kopecky will present on the life after death working.
Hey everybody, I'm Kai Kopecky. I am the lead of this Spark group that we're calling Life After Death, basically looking at how dead organisms, specifically foundation species, influence living organisms, like living foundation species, but also community dynamics and ecological processes. We had our Spark working group just back in February, the first week of February, but this project actually has been kind of going on since I started my postdoc about a couple of years ago at ESIIL, which is the Environmental Data Science Innovation and Impact Lab based at CU Boulder. But I did my PhD at UC Santa Barbara working with the Maria Coral Reef LTER. So I kind of did a lot of work looking at how dead corals influence populations and communities of living corals. And then I thought, as a postdoc, it'd be kind of cool to see if I could expand that framework into looking at how those dynamics maybe translate across different ecosystems that are characterized by foundation species. So yeah, the focus of this project has been looking at what we call foundation species. These are different types of organisms. They can be plants or animals that form biogenic structures. They typically are the most abundant organisms within an ecosystem, or they dominate in biomass. And they primarily interact with other organisms, or have non-trophic interactions with other organisms by providing habitat or modulating environmental conditions, or flows of nutrients to basically create the ecosystems that we actually know and study. So when foundation species are impacted by mortality events and disturbances, they can leave behind these really prevalent structures, which we call material legacies. Um, these can be, depending on the type of disturbance and how widespread it is across an ecosystem or a landscape, these material legacies can be really, really prominent and pervasive in recovering ecosystems. And therefore, they can have really strong influences over ecological processes. So the goals of our working group were to kind of explore how material legacies of foundation species influence living foundation species, as well as the communities that depend on foundation species, both in marine and terrestrial ecosystems. And then, so that was kind of like the beginning piece of this working group that led up to our in-person working group, or the Spark group that met just a couple months ago, where now we're trying to figure out what types of factors determine the strength of these legacy effects on living organisms. And sort of the last thing we would kind of want to do to button this up is take some of the lessons that this lessons and insights that we've gained from all these efforts, these synthesis efforts, to develop some like actionable guidance for managers and conservation practitioners for how to most effectively leverage material legacies in management, conservation, and restoration contexts. So, like I said, we kind of got a little bit of a head start with this project. We started working on this basically like this is how I got my postdoc at ESIIL was writing up this initial proposal at the end of my PhD. So I kind of hit the ground running a little bit with this, and we actually conducted a whole initial synthesis before our Spark group ever met in person. And what we really focused on is how material legacies affect demographic processes in living foundation species. And this is really different when you look across different ecosystems. So, for example, in forests, after a fire happens, standing burden trees can release seeds and stabilize topsoils that sort of facilitate the establishment of new seedlings afterwards. And therefore, standing burn trees actually promote recovery of a forest after a fire occurs. But then if you look in other ecosystems, some of the work that came out of my PhD, we showed that after something like a marine heat wave that causes coral bleaching, there can be these dead coral structures left on the reef. These actually serve as really good habitat for competing organisms, macroalgae or seaweeds, that actually they colonize the dead skeletons and then they can start to spread onto living corals that are still remain on the reef. And these seaweeds can actually take over. So in this case, the material legacy actually breaks down really key processes that are needed for corals to recover and therefore reduces the growth and survival of living corals. So just looking at these two prominent but completely conflicting examples from the literature, I was really interested in looking in a more general sense at how material legacies influence demographic processes in living foundation species, both in marine and terrestrial ecosystems. So we had an initial list of 10 ecosystems or LTER sites, and then we recently added in an 11th one. That's the Sevietta, which is why it has a dashed line there. But yeah, we have pretty much equal representation of terrestrial and marine ecosystems in our synthesis. And these span from the South Pacific with the Maria Coral Reef LTER up to the tropics in the southeastern United States and the Caribbean with Luquillo and Florida coastal ecosystems, and then a bunch of ecosystems and sites all in between that sort of span across the continental United States, and then all the way up to subpolar latitudes with Bonanza Creek. So we got a pretty wide swath of ecosystems we're working with. Sort of the thread that connects them all is that they're all really characterized by dynamics surrounding foundation species. And also, sort of the reason I ended up with this final list of sites was that that these sites all capture time series of both living and dead foundation species. And sort of that second part is the real key piece there. We needed to be able to have data where dead foundation species are measured in the same sort of sampling units, transects, plots, quadrats, or whatever, as the living foundation species so that we could directly estimate the effects of the dead on the living. So once we had our list of ecosystems and sites, and I should say here actually, we tried to use the kelp forest LTER site, Santa Barbara Coastal. The data structure were not quite lined up with how we wanted to ask that question, so we used sort of a cousin of the SBC LTER, which is the San Onofre Nuclear Generating Station Kelp Forest Monitoring Program. It's basically similar area in Southern California, and it's a long-term kelp forest monitoring site. Sorry, it's just right here. So once we had our list of sites and ecosystems, we started identifying, okay, what are the important foundation species we want to look at in each of these? What are the material legacies that they leave behind? And I should say here, this is not an exhaustive list of the foundation species or the material legacies. And then we also identified a demographic response that we think would be most strongly affected by the material legacies left in that system. And once again, the demographic responses here are not exhaustive with respect to the foundation species that we're studying. We just kind of wanted to key in on a few really important relationships between material legacies and foundation species demographic rates. And so we actually used both experimental and observational data here. Basically, whatever was the most appropriate for answering the question. In some cases, that was just long-term time series that had both living and dead foundation species. In other cases, it was long-term experiments where dead foundation species had actually been directly manipulated. And so we have like time spans that range from about 2 years all the way up to 32 years. So this first synthesis revealed some pretty striking patterns. When we look across these different ecosystems, and just to orient you on the axes here, this is just looking at the effect size of that relationship between material legacies and the demographic rate. And this is sort of a normalized, standardized way of modeling each of those relationships based on z-scores, which is just the units of standard deviation. And so the first thing that really popped out to us is that material legacy effects are really pretty common across all these different ecosystems that we looked at. We found significant relationships, or nearly so, in 9 out of the 10 ecosystems we looked at, the only exception being the kelp forest. We also found that positive and negative effects of material legacies were pretty much equally represented across marine and terrestrial ecosystems, and just equally represented in general. We had roughly the same number of negative and positive effects. And then lastly, we also found that the ranges for positive and negative relationships were about equivalent as well. So this is kind of like the endpoint of this initial synthesis that we did. And as you can imagine, when you see a bunch of effect sizes like this plotted with a bunch of really different ecosystems, a lot more questions emerge than what we were able to answer in the first place. So that's kind of what the purpose of this Spark working group has been, is to start looking into what is driving the variation in the strength of these legacy effects, both across ecosystems, but also within ecosystems. So that's kind of the next step we're at here. And one thing that we're trying, one of these projects that's coming out of our working group is looking at what we're calling these spatio-temporal attributes of material legacies. And just to put that in more concrete terms, we're looking at how quickly material legacies are added to the environment. So organisms die in different ways, whether that's just kind of ambient background mortality or pulse disturbances. But that can kind of determine the rate at which legacies are being produced in an environment, whether it's gradual or really abrupt. Another attribute we're looking at is what we're calling the residence time. This is how, once legacies are produced in the environment, how long do they actually remain there? Because there are lots of different processes that are actively removing legacies from the environment, like decomposition of wood and leaf litter, or erosion of dead coral skeletons, burial of oyster shells, burning of grass litter, consumption of dead materials by microorganisms, lateral transport of plant material and marsh rack. So there are a lot of things that are actually constraining how long a legacy is residing within an environment, determining whether that legacy is ephemeral or persistent within its environment. And then the last attribute we're looking at here is just how spatially extensive are legacies distributed across a landscape. So mortality and disturbance events are not always completely uniform and homogeneous across an environment. Sometimes it can be really patchy, or there can be just certain interfaces where mortality happens most strongly. Therefore, legacies are not always spread across an environment completely uniformly, and probably the extent at which these legacies are laid out on a landscape is going to determine how strong their effects are. And so we're going to try to, for as many of these ecosystems as these LTR sites as we can, quantify these different axes, these different attributes, and then get sort of an estimated effect size based on these three attributes. Then we'll go back and map how these predicted effect sizes um, align with the original effect sizes we found in our initial synthesis. And so this is hopefully going to be able to explain a little bit of that variation in the strength of effect sizes. Again, both across ecosystems and as well as within ecosystems. So the other big piece of our working group that we're looking into is how do these effects on foundation species translate to community level responses? So again, looking back at that initial synthesis, we found several direct effects of material legacies on living foundation species, both positive and negative, or facilitative and inhibitive effects um, through these various mechanisms listed here. So now what we're trying to do is look at how either facilitative or inhibitive effects on living foundation species translate to more indirect follow-on effects on overall community structure. So you can imagine when foundation species or when material legacies have facilitative effects that drive recovery of foundation species, that might help stabilize the original community structure that existed before a disturbance or mortality event took place, and therefore it might maintain the original community structure that existed. On the other hand, when there are inhibitive effects, when legacies prevent foundation species from recovering, this actually might cause the system to veer off towards an altered or degraded community structure after a disturbance that produces material legacies. And so what we're trying to achieve here is actually more of a predictive modeling framework. First, we want to build a very general model structure where we look at foundation species, both at early and late life stages, and how they're affected by material legacies. And what we have here as this intermediate step um, is an abiotic condition that is affected by the material legacy, whether that's light resources, nutrient pools, structure for habitat, different things like that. So we're going to model how material legacies are affecting an abiotic condition that then it translates to effects on both foundation species. Then these other bubbles up here are either what we're calling mutualists on the left. So other species in the community that co-vary positively with foundation species, or competitive species, which we have listed over here on the right, which are other species in the community that co-vary negatively with foundation species and might compete for the same resources. So from this general model that sort of captures all the possible dynamics and pathways, we're going to break it down into submodules that actually reflect realistic ecosystem dynamics that can map onto each of our LTR sites. So an example would be what we're calling resource modification, where a material legacy has some positive effect on an abiotic condition. For oyster reefs, that could be physical structure for settling oyster larvae, or in temperate forests, that could be nutrients from decomposing wood that help stimulate tree growth. And therefore, that material legacy is going to have a positive effect on that abiotic condition, which then has a positive effect on the foundation species, which helps that system recover or helps the foundation species recover. But then we add some of these other communities, species into the mix, we might have different dynamics unfold. So in this example, where we have a mutualist that is positively affected by the material legacy, um, this might actually have an added positive effect on the foundation species, where it might drive an even faster recovery. But then on the opposite side of things, when there's something like a competitor that's facilitated by the material legacy, that's going to have a negative effect on the foundation species. And therefore, you might have something like a regime shift take place where the competitor is able to consume that material legacy as a resource and basically take over. So these are just kind of some examples of like submodules of that general model and some cartoon dynamic trajectories for how things might unfold after a disturbance event. So finally, what we want to do is we want to take all these insights that we've gained from our initial synthesis, from our follow-on studies, and even from some further literature review, and sort of assemble a bit of some guidelines for how material legacies might be able to be effectively incorporated into management, conservation, and restoration efforts. This is already done in several different ecosystems. For example, in forests where they cut down dead trees so that they can decompose and release nutrients into the soils, or on oyster reefs where they deploy dead shells to serve as hard substrate for new oysters to settle on. On coral reefs, some new work is showing that you can stabilize or even completely remove dead coral skeletons from the reef to sort of prevent macroalgae or seaweed from colonizing and give corals a better chance at recovering. Or in grasslands, where they burn off dead grass litter to make way for new growth, new grass growth to occur. So there are already a lot of ways that people are utilizing or leveraging material legacies for in these different contexts, but we're going to try to come up with a little bit more of a general set of guidelines that can apply maybe beyond these ecosystems where this is already practiced, to show that this is actually a really important thing that you could be thinking about if you're trying to foster desired outcomes in this really nature-based way. And so with that, yeah, that's all I have for you today. So I'll take any questions.
Awesome analysis, Kai. Really appreciate it. Um, we're going to open it up for questions. I will actually jump in first, I guess, and ask. 10 ecosystems is terrific, but I can imagine you might be running into a power problem as you uh, try and model three different um, additional factors. Have you thought about where else you might be able to reach out to for additional data sources?
Yeah, like other LTER sites.
Other LTRs, well, international LTR sites would be one great opportunity, but other sites as well that have longer term data about a variety of foundation species.
Yeah, I mean, I think we're definitely open to that.
Okay.
Um, and especially now that I have like, this really awesome team within this working group who's kind of like helping manage the load, and also is just like a really good everyone's got really good knowledge of like where to find other data sets. So we're definitely interested in like incorporating more. And anyone on this call who feels like they have long-term data that might fit into this framework, yeah, we're totally open to that. Now that we have the capacity to ingest more.
Right.
Yeah.
Cool. Other questions for Kai? Kai and Ty, I guess. Katie couldn't make it, but
Yeah, Max is here as well.
oh, great.
As well as a couple other members from the group. So feel free to direct
Awesome.
questions at any of us. Or not.
All right. Um, Very cool. I'm excited to see where the next steps take you.
Yeah, thanks, Marty.
Um, all right, so we will shift gears now to Shalanda and the Taxonomic and Functional Diversity Working Group team, which did not have the advantage of Kai's postdoc leading up to the working group, but are still doing really cool work.
So, thanks so much for your great presentation, Kai. Super interesting work you all are doing. Yes, so I'll go ahead and get started. So we were a group funded by the Spark, and we had very big aspirations. So we're really interested in the temporal variation of taxonomic and functional diversity and how this impacts ecosystem functions such as nutrient cycling of consumers across aquatic ecosystems and LTR sites. And we are terming ourselves Consumer Functional Diversity Working Group. So this is the group. No one could be here today. Some folks are in just dispersed all over doing different tasks. So I am the sole person here today, but here's our amazing team. We have folks from graduate students all the way up to assistant and full professors. So a nice mix of folks. So thank you already to the whole team that has been helping with this effort. Also, I guess I didn't introduce myself. My name is Shalanda Grier. I'm a postdoc at UCSB in the Berkey Powell Lab. So we really built our working group off of our prior working group's data product. So there was a consumer nutrient dynamics working group, and this was led by Mac White. And they really worked on estimating excretion rates for fishes across different LTR sites. And so we thought it would be a great idea to take advantage of this product that they created and utilize it to better understand how ecosystem functions are changing within ecosystems among consumers. So one thing that was really interesting that came from this working group that is now a paper that White Mac has submitted is that we found that, species richness and species asynchrony are strongly explained by variation in excretion rates. So excretion rates, changes in excretion rates we saw very closely aligned with species richness, as you might imagine, and also how different species change in their abundance at different times. So that was really interesting. And we wanted to take it a step further to just think about different relationships that we can find within an ecosystem as it relates to ecosystem functions such as excretion rates. So we want to know how do changes in consumers impact this ecosystem function, and specifically, how do functional traits impact changes that we see in ecosystem function. So in the literature, we see a lot of debate now whether or not we should just think about species identity solely versus also thinking about functional traits. And we see this butterfly fish here, and we can think about a functional trait as any type of characteristic, whether that be morphological, physiological, behavioral characteristic that impacts an individual's survival, growth, or reproduction. So we were thinking, instead of thinking about species solely, let's think about these traits that we know have direct relationships to the overall fitness of an individual, and how does this relationship then thereby impact the ecosystem functions that we see. So these can give us better insights about into the mechanisms that drive changes in ecological processes and just overall trends in communities and ecosystems. And so we've seen much success of folks using functional traits in across different disciplines and systems, but specifically, I think people are very familiar with this in the terrestrial world when we think about plants and we think about frameworks such as the leaf economic spectrum that kind of groups individuals on this continuum of whether or not they quickly acquire resources, are more conservative with their resource requirements and uptake of resources, and how these then thereby influence different ecological processes such as leaf composition rate or root decomposition. But we've also seen people use functional traits in other systems. In addition to plants, this spectrum is this framework, this leaf economics framework is used across different ecosystems with plant species, but we also see it with algae traits. We also see it in vertebrate communities and also with fishes as well and birds as well, different vertebrate communities of people utilizing these functional traits to understand broad scale patterns. However, looking at these functional traits across taxa is a little bit more rare. And so we have had recent advances looking at cross-taxa studies, so looking at taxa that are from very disparate lineages to see how we can use universal traits to understand how different taxa are changing together. And so some work has been done trying to understand this. This is a figure from Junker et al. 2022, where they took terrestrial species from 7 different classes and they assigned what they are calling universal traits. So these traits were reproductive rate, age at first reproduction, lifespan, offspring mass, and metabolic rates. And they were trying to understand how do individuals from these different classes map onto trait space, and if there are any type of ecological trade-offs that we can see. And they were able to see that similar to the leaf economic spectrums where there's this trade-off between like this fast and slow return, that there was also a trade-off among these terrestrial species, specifically between reproductive strategies and life history strategies. And it was very similar to what we kind of already have thought of and like a lot of seminal work with like life history traits of fast and slow continuums. So that was really interesting. And this really motivated me and encouraged me to think about like how might this apply to aquatic species across taxa that might be a little bit more challenging, but I think worth the reward to understand these patterns as well. And so initially our proposal, we wanted to have a conglomerate, if you will, of consumer groups and look at what we would identify as unifying traits based on these categories from this Junker et al. Paper, where they looked at organism size, biological times, biological rates, feeding characteristics, and circadian rhythm. And then we wanted to take these individuals from all these different consumer groups and map them onto trait space. So that is the figure you see in B. And to understand like how do these individuals group together, are how variable are they within the trait space, and if they group by ecosystems and if they group by actual consumer groups. And then we wanted to understand how does the functional diversity, so when we look at the differences in traits dispersed within a community, how does that then impact our explained variation that we see in ecosystem function? And we had another big aspiration of thinking about this in terms of thermal disturbances and just disturbances or variation in these metrics over time with across the different LTR sites. So as you imagine, this is a very big like goal that we had, and we don't have much time, but we were able to do a lot in the time that we've been allotted. And I think have come up with some really interesting or we've seen some really interesting trends and patterns that we are going to delve more into today. So I'm excited to share those with you today. But we recently had our in-person working group in March. And from that working group, we kind of reformulated our questions a little bit. And so I'm going to share those with you today. So as it relates to trait space, we wanted to kind of create a standardized trait space. So find unifying traits across all the consumer groups that we can locate or identify, and understand how does shifts in the aggregation of traits change through time, and are these predictable? And within any community, do we see a certain amount of biomass of traits being represented? So do we see a heavier biomass of higher trophic levels or lower trophic levels throughout these consumer communities? And are they the same or are they different? And do we have these hotspots? Are they shifting similarly through time? These hotspots of biomass, they change the same through time. So that's like our first paper and um investigation that we're interested in. And the second investigation or paper that kind of developed or came out of our working group was thinking about excretion rates and how these fluctuate through time for consumers. And we are focusing on right now on bird, fish, and zooplankton consumers, although we started out with a large list of consumers, and we'll talk about that a little bit more. And under this question too, we're trying to understand how do excretion are whether or not species richness or community weighted mean values best predict excretion rates. So community weighted means are just like if you have, it explains the dominance of traits within a community based on abundance. So if you have a lot of individuals in a community that have, let's say, a higher body size or a larger body size versus a smaller body size, then that trait value will be more represented in the community weighted mean. And so starting out, we had many LTR sites. We had terrestrial and aquatic. And initially, when we wrote this proposal, we were focusing on aquatic, but as we continued to meet with one another, we got really interested in also adding terrestrial sites. So that came our big list of consumers. So we had quite a nice representation of consumer groups for mammals, insects, birds, and also reptiles. And then our aquatic sites were mostly just fish and zooplankton, of course, and a nice representation of many LTR sites and ecosystems as well. And so two of the data products we want to get from this working group is to have our own nutrient excretion estimate data product with new sites that we added onto the C&D working group, and to also have our own trait database. So we had two different workflows. The first being just supplementing or adding onto our original nutrient excretion estimates. And so those estimates mostly looked at MCR, FCE, SBC, VCR, and PSCO, Central and South. And so we added new sites to have a bit more coverage so that we can have some freshwater coverage, and that we can also have a little bit more coverage with tropical ecosystems, specifically coral reefs. So we added Arctic LTR, Palmer, North Temperate Lakes, and for the fish consumer group, we added Maria Island, Brightness Island, and Nigaloo Reef, and these came from the Reef Life Survey long-term monitoring program. And so our first data product and something that we're almost done with is finalizing these excretion estimates from these new LTR sites onto the C&D so that we can have our own CFD product of excretion rates. And we calculated these excretion rates using coefficient estimates from Bonnie and McIntyre that were largely based on diet of all these consumers. So whether they ate algae, fish, invertebrates, or fish and invertebrates. And of course, the coefficients increase depending on the trophic level or the diet of the individual. And so our next workflow that we worked on a lot prior to our in-person working group meeting and also during our working group meeting was just compiling all this trait data together. So we have more than, I think, 12 trait databases that we pulled trait data from to have this centralized functional trait database that we're really excited about and that other people will be able to use across different LTR sites. And so we had fish, amphibians, zooplankton, small mammals, insects, and birds. And we looked at an assortment of functional traits, or we tried to collect an assortment of functional traits ranging from mass, length, weight, lifespan, maturity, reproductive rates, metabolic rates, activity times. And so it was a great big effort that required a lot of time from a lot of folks on the team. And so huge shout out and thank you to everyone on the team. But of course, as you can imagine, some data was sparse, some data was very abundant and available. And so we went through trait coverage to see like after imputation, so after filling in missing trait data, like how much coverage did we have? And we saw that we still didn't have, we had a decent amount of coverage actually for what we were collecting, but there were still gaps in our data. And so for instance, for our insect data, we only had like 2 traits that had, more than 50% coverage. So we had to make a decision about which groups we would use and which traits we would actually use moving forward, just to have the best return on our investment. And so we essentially decided to use fish, so plankton and birds. And we thought that these would be very interesting groups to compare to one another and to think about in trait space. They're very different. They span very different types of environments and ecosystems. So we thought that we could still get some interesting results from this. And so the functional traits that we decided to focus on were lifespan, age at maturity, reproduction, length, and trophic level. And as you can imagine, during our working group, we created many different figures and had a lot of great discussion. And so I'm just going to highlight some of the things that came out and kind of talk about where we are hoping to go next. So the first big task that we had was to create a trait space and to make sure that our traits were homologous, that they are comparable across our consumer groups. And so we did have success with that, and that was a big win and very exciting for us that we were able to create a trait space, and we saw groupings of individual organisms or individuals within this trait space. But all of these figures I'm going to show you are preliminary, as we're still like parsing out some of our data and cleaning some of our data, just like doing a lot of quality control. But this first image I'm going to show you is the trait space with fish and birds. And so our PC1 is mostly driven by mass and age. So we see our PC1 here. And our PC2 is driven by trophic level. And our PC3 is driven by reproduction. So in yellow, we have birds, and in green, we have fish. And we see that the individuals from both consumer groups are grouping into trait space based off their traits alone, which was very interesting. So larger body size individuals are grouping together. Individuals with from low to high trophic levels are grouping together. So that was really exciting for us to see that, okay, it worked. These traits are comparable, and we are able to create a trait space across taxa, which is really exciting. And so our goal now is to include our zooplankton traits and also our RLS data. There was some quality control we had to do. So we are expecting some of these to change, but initially we are just very excited that we're able to just create this space in the first place and that we see some strong variation across our groups. So something else we were really interested in is understanding. Initially, I mentioned we were interested in understanding like the density or the mass or the representation of traits within trait space and how that might change over time. And so we were able to look at the centroids, so the movement of the communities based off of their traits over time. And so these figures are essentially showing either from top down, so north to south, or east to west, left to right, how the centroids of each community, our program, we're calling them projects, are moving. So right here, we just have Peace Coast, Central and South, FCE, MCR, SPC, and BCR. So we're looking at how in terms of traits and in terms of biomass, how is the representation of traits moving across time? And so we were able to see that for MCR, and this is a coral reef site, that the centroid of this community moves from north to south. And this is largely explained by the trophic level. From south to north. And so from lower trophic levels to higher trophic levels. Over time, and this tracked with myself, who works with the MCR data, and other folks in our program, in our group who work with MCR data. We know that over time, we are seeing changes in trophic levels due to also changes in coral reef structure. So that was really interesting. And then we're able to see with the other projects, specifically PEACECO, FCE, and SBC, we saw a shift in the centroid from east to west. So body mass to lifespan, so from lower body masses and younger individuals to older individuals over time with that are also larger in size as well. So this was really interesting and something we want to continue to investigate, and adding the RLS sites that has another coral reef site will allow us to see whether or not we have similarities and patterns with the movement of north to south or south to north with the trophic level, similarly to MCR. So we're really excited about this, but we're also excited about adding our other consumer groups into this data. So initially, when we started this project, the only biomass data we had were for fishes and zooplankton. But during our working group, folks got really excited and we thought, like, what about the biomass of other species and not just their traits? So the figure I showed you before was solely based on traits and the similarity or dissimilarity of individual consumers based off their traits, whereas this is also looking at biomass and traits. So we have decided to add the biomass of other consumer groups, such as birds. So that'll be really exciting to see, okay, do we see similar changes? And are there any patterns that we can see across ecosystem types? Are they just solely or largely within projects? So our second question, where we're thinking more so about our traits, our community-weighted mean values, our functional diversity, and our excretion, we really wanted to understand, like, how do these metrics relate to excretion rates and the variation in excretion rates that we see? And so, as you can imagine, we also have so many figures that I could show you. I'm just going to highlight two today. But the first thing that we did, we wanted to look at these correlations with our main traits that we decided to visualize our focus on, which was lifespan, maturity, trophic level, and length. And I'm just going to show you today biomass and lifespan. One is just like a gut check to make sure that, like, okay, our data makes sense. And the other is just to show you a cool pattern that we saw and that we hope to investigate some more. So when we look at excretion rate, which is on our y-axis, and community biomass of these projects, we see what we would hope to see and what we would expect to see, that when you have more biomass, essentially you have greater values in excretion. So you have more of that ecosystem function of that nutrient cycling there. Well, is a proxy for that. And this is solely across fish projects again. And so something else we're working on is finalizing our zooplankton. So we can also look at this with our zooplankton species and also our bird species as well. But this is really just a gut check for us to make sure that, okay, our data is aligning and doing what we think it should do. And this is more of an exploration. So when we looked into other community-weighted mean trait values, such as lifespan, we start to see some interesting patterns. So for example, when we look at excretion and lifespan, we see that there seems to be a positive trend, specifically among the PEACECO sites, that when individuals are older in age, that they tend to excrete more. And all these dots are just site and year combinations. So for each project across all the years of the survey, that's what those dots represent. So this was really interesting. We are just starting to really just look at our data and then think about, like, a different analysis we will run. But this was something that we got really excited about because, of course, you have so much data and you don't always find a pattern. So this was a pattern that we thought was interesting. And we know that at PEACECO, there are a lot of longer lived individuals. And we saw that with some other figures and data that we explored within our working group. And so some of the next steps and future plans we have for our data exploration and just preparing our data are to, for our bird data specifically, we want to continue to visualize the movement of trait space, like I said earlier. So when we're looking at the change from, like, east to west or north to south, we want to also do that with our bird data. And so we are adding those biomass data from bird study varies from 7 additional LTR sites. And someone else in our working group is looking to try to work on calculating bird excretion rates. There are different models. We're also trying to talk to bird people who are more just experts than we are to help us make sure that we're thinking about these bird excretion rates right and that we use the right models. So that's something that we're doing and we're really excited about. Something that we're almost done with is just completing our CFD excretion package. So we needed to go and mine more zooplankton traits, specifically dry mass, so that we can finish our calculations and really finalize our estimates of nutrient excretion rates, and then find other functional trait data such as lifespan. But we're almost done with that. And we think with that in our imputation process, we should have pretty good coverage for our zooplankton. So we'll be able to add it into our workflow, add it into our trait space, and continue on with our workflow. And we also plan to reach out to a zooplankton expert. We already have one who we will hopefully talk to shortly. And then lastly, again, we just want to add all our consumers into the same workflow. So we're seeing some really interesting patterns with like consumer weight, community weighted means and excretion rates. We also want to see if we can see similar things with the zooplankton and birds. So we just want to add all our consumer groups together so that we can start seeing or visualizing these trends across consumer groups to see if there are patterns across groups, ecosystems, are these patterns just mainly within programs. And lastly, something else that we're interested in and we plan on doing is thinking about species richness and community excretion. So we're thinking about understanding how the removal of certain species from key trophic groups, how that then influenced excretion. So kind of do like a stepwise removal and to see like over time, like how is this impacting our excretion rates. So these are things we're excited about working on this summer and right now. And it's a lot of work ahead of us, but we're really excited about some of the preliminary results and figures we've been able to produce. And with that, I will take any questions. And I also just want to thank NCs and the LNO and Marty and Ginger specifically for all your help with the working group logistics.
Thank you so much, Shalanda. Great talk. Do we have questions from the group at all? I had super quick, just a clarification question about the flower plots.
Okay, yeah.
Those are, I don't think you need to, well, you can bring them back up if you want. The movement that you're talking about is through the trait space, right?
Yes, yes.
And each of the flower petals is a year or a measurement sequence, or what do they represent exactly?
Yeah, this is showing the size of them as the biomass, and it's just over time, how much biomass is being is from over time. So sorry. Yes, south to north is where it starts and then it ends, and east to west is where it's starting and ending.
Okay.
Yeah. So just the size of them is just like the biomass.
Okay. I'm still not completely clear on how time is represented there, but
It's not like explicitly represented here.
okay.
This is mostly just like the movement of biomass.
Got it.
Mm-hmm.
Okay. Cool. Thank you.
Yeah.
All right. Well, much appreciated, both of you. We will have the recording up and available to share with other folks within a couple of days. And I imagine both Shalanda and Kai are available to contact if you have additional questions or ideas for data to bring into the analyses. That'd be great. Thank you to everyone.
Thanks, Marty.
Thanks, Marty.